Hi everyone, so this is the third genetics video in the genetics video series, and this video is on different inheritance situations and genetic disorders. So in the last video we talked about the structure of DNAs, alleles of DNAs, and how you get your looks from your parents. Well, in this we're basically just going to go a little bit more in depth with that. So let's get into it. Epistasis. So I know that specifically previously we discussed about how there are two alleles on the chromosome, one from each parent, and how this determines your own phenotype. But there's also a phenomenon in which the phenotypic expression of a gene, the way it basically expresses a gene and how it looks, at one location will alter the gene at a different locus or location. This is known as epistasis. For instance, in labradoodles, there is one gene at the first locus that determines if the dog is black or brown, and the gene at the second locus determines the pigment. So for instance, big E or big E small e says that either a brown or black pigment will be given, but small e says that a yellow pigment will be given. And if the second locus gene is small e, and the first locus gene is dominant for black or big E, then the dog will still be yellow because of the second locus kind of acting on the first locus. So both of these genes are affecting dog hair or dog color hair, and so this is known as epistasis. Polygenic and Pleiotropy. In addition to a second gene being affected by the first, and thus the phenotype being changed, which is what happens in epistasis, there is also a type of gene inheritance in which two genes together affect a single characteristic. This is called polygenic inheritance. The opposite of polygenic inheritance is pleiotropy, in which one gene on the chromosome affects many traits. So for instance here, this one gene on this chromosome affects both the eyes and the heart and many other facets of your body. And in case of polygenic inheritance, it's taking two genes and it is linking them to one character. So now that we've talked about different inheritance patterns, we're going to move on into the diseases you can actually inherit. So you've probably heard the phrase, "it runs in the family." Well, this is because a certain mistake in one of your ancestors' genetic code was passed down generation to generation, and it was continually expressed. For instance, if your great-grandfather had a disease and it was passed down to your father, and that was passed down, or your grandfather, that was passed down to your father, and that was passed down to one of you or your siblings, then that means that this is an inherited gene, and there was a mutation in one of your ancestors' genes, in this case your great-grandfather, and this was continually expressed and passed down. But luckily, most of these genetic orders are inherited as recessive traits, so only people with homozygous genes will express the trait. Homozygous recessive genes is what we're looking for, like RR, small r, little e, anything that's lowercase or homozygous recessive. So, some examples of the actual diseases that are common are cystic fibrosis, which is actually the most common lethal genetic disorder in the U.S. It's basically when the recessive allele has an absence or defect of a chloride transport channel. And these channels basically carry the chloride ions to the extracellular membrane, but since there is a defect, it causes the ions to not be able to be transported, and it causes them to build up, and it creates a thick membrane or mucus that surrounds the cell of the intracellular chloride, and it basically allows the cell to be thicker, the mucus around the cell to be thicker, and this basically inhibits organs to perform their actual task. There's another disease called sickle cell disease, and this is the most common lethal disease in African Americans, and this is basically when a single amino acid in hemoglobin, or your red blood cells, is substituted for a different amino acid, so kind of think about it as point mutation, and it occurs in homozygous individuals where all the cells in the hemoglobin become sickle cells or like abnormally shaped. And although the genes need two recessive alleles to actually have or express the disease, at the organisamal level, the normal allele is incompletely dominant, and so even with one abnormal allele, this can still affect the phenotype and the cell can still be a little bit abnormally shaped. But at the molecular level, there are codominant, so again, it's still affecting the phenotype. So this one, it can still affect how the cell is actually shaped, even if there's only one dominant allele. So on the other hand of the recessive disorders that we just discussed, with the homozygous recessive alleles being present, and this causes the expression of the disease to be shown, there's also dominant disorders. Now, these disorders are usually very harmful and almost always result in death. And if you have a dominant allele from each parent, then it will most certainly result in death. So if you're homozygous dominant, then it'll result in death for this disorder. But luckily, these disorders are very rare as compared to recessive disorders. And this is actually because the diseases are like this are likely to develop early on in your life. And those who die because of these dominant disorders or diseases, they die out and they're not able to reproduce or pass these genes on to their offspring. So this causes them to kind of like die out, and which prevents the passing on of these mutagenous genetic codes. So one example of this is Huntington's disease, and there's also achondroplasia, which is a form of dwarfism where people are shorter than they usually should be, and this is very rare, occurring in only about 99.9% of people actually don't have the disease, so it occurs in only like 0.1 or 0.1% of the population. So branching out from diseases, and disorders, there's also multifactorial disorders. So this is kind of a combination of the genetics inheritance that we've been talking about, where there's a mutation in someone's gene and this is passed down, and it's expressed or it's not expressed. And it's also a combination of environmental factors, such as not in your actual genes, but because of how your lifestyle is, it can cause a disorder. So some multifactorial disorders are diabetes, alcoholism, heart disease, and many of the diseases that are multifactorial are usually cardiovascular diseases. And so let's take diabetes, for instance. It might be partly genetic in that you're predisposed to having the diabetes, but it can also mean that you've just been consuming a lot of food that's going to also predispose you to having diabetes. So both of these factors into consideration can cause multifactorial disorders. So like I was talking about earlier, how the dominant disorders, they're not seen as prominently because the people that do have it usually die at an earlier age and they're not able to reproduce. And this all sounds very scary, but luckily there are measures that can be taken so that we are aware of the diseases in the baby. And this is through fetal testing. So one kind of fetal testing is called amniocentesis, and this is basically when there's a needle and it's injected into the amniotic fluid, which is basically a fluid that dampens the fetus at the uterus. And genetic disorders can be found through this method. Another type of fetal testing is chronic villus sampling, or CVS. So this is a method which is conducted by a physician who inserts a tube running through the cervix and through the uterus, which then takes a piece of tissue from the placenta and this tissue is important as it absorbs the nutrients and waste of the fetal from the mother, and the cells of this tissue can be created rapidly, so the cells can be tested on. And they take this tissue and they analyze it for, again, all the things that have been coming out, and this can also lend itself to see if there's any disorders of the baby. And the third kind of fetal testing that we'll talk about is ultrasound. So this does not involve any needles or tubes, which might have sounded kind of intrusive and painful even in the first two methods, and it instead uses sound waves to create an image of the baby in order to find if there may be any disorders that appear visibly. So there's also something called newborn screening. So instead of testing the baby while it is still in the fetus or in the stomach, some genetic disorders can wait to be found after the child is born. So these tests are usually biochemical tests. So a baby will usually be tested or screened when they're first born in order to check for pneumonia or PKU. And this is basically a disorder in which the baby cannot correctly metabolize the amino acid phenylalanine, and too much of this in the body can give the child mental disorders. So fortunately, finding this at birth can help to avoid these problems because we can instill a diet for the baby and it can thus become cured and healthy. However, the curation is not very likely, but luckily we can try and take steps towards this. So that was it for this video. So that was it for this video. I know it was kind of confusing with all the different kinds of disorders, inheritance, and testings that can occur. If you have any questions, then please leave them in the comments below, and subscribe to this channel if you want more of these videos on genetics, because I definitely have more coming up. And feel free to watch the previous genetics videos, because this is a series. And we have a bunch of other fascinating videos on this channel, so go ahead and check those out too. Here are a couple of citations I used for this video. So here are a couple of more citations I used. And here are the last bit of citations. Here are the last bit of citations I used for the pictures for this video. Again, feel free to ask any questions or comment down below about future videos, ideas that you might have, and subscribe to this channel. Thanks, bye.